Good evening, everyone. At the outset, I would like to thank the organizing committee for inviting me to talk on this important topic of radiation biology of SABR for spinal metastasis. Treating spine mets has undergone a major paradigm shift in the contemporary radiotherapy practice, with SABR delivering very high doses to that of the spinal mets. And this has been possible due to major evolution in the radiation delivery technologies. However, one major limitation still remains, that is radiation-induced myelopathy, and this is because of the proximity of that of the spinal cord to the uh, target organ. Therefore, harnessing the radiobiological aspects of high radiation doses to the tumor and sparing the spinal cord remains the key to the success of therapy. Typically, in SABR or in SBRT for spine, we give radiation doses in the range of 18 to 24 gray in single fraction or 25 to 30 gray in 2 to 3 fractions. This is done because the radiobiological principles are exploited in SBRT, wherein small volumes of normal tissue can withstand high doses of radiation with resultant low complication rates as opposed to large volumes of normal tissue. So, what is the radiation biology of SABR? And what is the impact of the 5 R's of radiobiology? That is repair, redistribution, repopulation, oxygenation, and radiosensitivity on SABR. So thus, the 5 R's of radiobiology hold true for SABR. Coming to repair of sublethal damage, SABR, or for that matter SRS, lasts for a considerable length of time, so that repair of SLD still happens when we are giving SABR doses. And this has been shown in vitro studies by Fowler and Ling also. Therefore, the estimated loss of biological effect due to SABR is about 10% if the fraction time goes beyond 30 minutes. Therefore, it is always prudent to reduce the fraction time less than 30 minutes so that we can reduce the repair of SLD of tumor cells. What about hypoxia and reoxygenation of the surviving tumor cells? We all know that in conventional fractionation, reoxygenation happens. Reoxygenation of that of the surviving clonogenic tumor cells occur till a dose of 10 gray. However, at doses beyond 10 gray, hypoxia and acidic microenvironment prevents reoxygenation, especially in tumor cells which are in phase of chronic hypoxia. Therefore, at high doses, indirect cell death occurs due to massive vascular damage. And in single fraction SABR, reoxygenation of tumor cells therefore becomes irrelevant. What about repopulation or regeneration of the surviving clonogenic tumor cells? The biologic significance of accelerated repopulation after SABR is not fully understood. We all know that following conventional fractionation radiation, there is accelerated repopulation after second or third week of radiation therapy. However, for high dose single or multifractionated treatment, which is completed within a week's time, compensatory repopulation during the treatment when we are giving high radiation doses in SABR range is negligible. Hence, delivering a high dose of radiation may mitigate clonal expansion of the tumor cells, thereby offering a significant advantage to SABR. Coming to redistribution of the tumor cells within the cell cycle, when we are reducing the number of fractions, this alters the probability of irradiating a cohort of tumor cells as they move into the radiosensitive phase of the cell cycle, which is the T2M phase of the cell cycle. Once we prevent the cell cycle progression, or for that matter, a cell cycle arrest happens, this leads to interphase death, occurs because of apoptosis and necrosis, but also because of tumor cell senescence and autophagy. What about individual tumor cell radiosensitivity? SBRT nullifies the differences in the tumor death that arises due to the variation in tumor cell radiosensitivity. Therefore, in SBRT, when we are giving large doses and short treatment time, it provides less opportunity for the resistant stem cells to develop. So, cell death with SRS or SABR cannot be just explained by the 5 R's of radiobiology, which we see during conventional fractionation radiotherapy. there is something more than 5 hours which are involved. First is the endothelial cell damage that may enhance the cytotoxic effect irradiation on the tumor cells, and this is because of the endothelial cell apoptosis. Vascular damage at high doses produces secondary cell killing, and doses higher than 10 gray induce this vascular damage, leading to indirect tumor cell death. Apart from the vascular damage, which is the resultant of endothelial cell apoptosis, there is also enhanced anti-tumor immunity after high-dose tumor radiation. Last but not the least, some small in vivo studies have shown abscopal effect when we are giving radiation doses in the SABR range. Thus, to summarize the mechanism of cell death after SABR, tumor cell death can happen as a result of direct tumor cell kill. And this is not the only mechanism. You can also have indirect tumor cell kill with SABR doses, and that can be because of vascular damage and immune responses. So what happens in radiation-induced vascular damage? When we are giving radiation doses on 10 gray, vascular damage happens by endothelial cell apoptosis, which I've already explained in my previous slide, and this is followed by tumor cell death. Now, endothelial cell apoptosis can be induced via two pathways, and this is very much dose dependent. When we are giving SABR doses of more than 10 gray, it is the sphingomyelinase pathway which is responsible for endothelial cell apoptosis. When we are giving radiation doses beyond 17 gray, it is the ceramide pathway which is responsible for endothelial cell apoptosis. So apart from these two mechanisms for vascular damage, extreme hypofractionation or high doses of radiation, which we give in SABR range, damages the perivascular niche, which contains the radioresistant stem cells. So when we give very high doses of radiation, what happens to the blood vessels? These blood vessels are considered as serial architecture structures. Injuring a single focal point of the blood vessel by a very high dose of radiation may result in obstruction or may completely halt the downstream blood flow, resulting in a cascade of tumor cell death, which reside in the defunct vessel. For theoretical purpose, a single endothelial cell subtends or provides nutrition to a segment of tumor cells containing 2000 tumor cells. Imagine the small endothelial cell undergoes apoptosis and cell death can result in death of about 2000 tumor cells. So this is a cartoon which shows the difference in the tumor cell kill mechanism when we are giving conventional fractionation radiation and very high radiation doses in the SABR range. When we are giving 2 gray radiation in fractionation, there is no damage to that of the blood vessel. What is happening is that there is reduction in that of the oxygen cell content, and the hypoxic cell undergoes reoxygenation in the next fraction, and it undergoes cell death. But when we are giving very high doses of radiation in the range of 20 gray, what is happening is that it is causing damage to that of the blood vessel, but nothing is happening to that of the hypoxic cells. And the tumor cell death is actually happening because of that of the damage to that of the blood vessel and the perivascular niche area, which contains the stem cells. So this is a cartoon which shows the molecular response to radiosurgery. On the left-hand side panel is the tumor stem cells. When we are giving radiation doses beyond 10 gray, there is a double-stranded DNA break, resulting in post-mitotic cell death or also increased in the enzyme called as ceramide synthetase, leading to increased production of ceramide, which leads to apoptosis. On the right-hand side panel is the vascular endothelial cell. When we are giving radiation doses beyond 10 gray, it causes activation of acid sphingomyelinase enzyme, leading to increased expression of sphingomyelin, ultimately resulting in increased production of ceramide, which leads to increased production of, again, pro-apoptotic proteins called as Bax protein, and ultimately leading to endothelial cell apoptosis. When we are giving radiation doses beyond 17 gray, there is activation of the ceramide synthetase enzyme, leading to production of ceramide and ultimately leading to the apoptosis. So the bottom line is the tumor cell death when we're giving high radiation doses can be because of endothelial cell apoptosis and DNA damage, which act together both in concert with each other to produce tumor cell death. And this is called as a two-target model. So what is the hypothetical cell death mechanism by SBRT? Conventionally, a tumor will have about 15 to 20% of hypoxic cells. Going by the classical radiobiological principle, the cell survival curve is considered to be biphasic. For representational purpose, you can think of curve A represents the cell survival curve due to the cell death of that of the toxic cells, wherein you require only 0 to 5 gray to bring about cell death. But the curve B, which represents the cell survival curve due to hypoxic cell death, requires quite high doses between 5 to 12 gray to bring about cell death. But practically, what happens is that we see curve C and curve D at high doses, that is at around 12 gray. And this is because of the vascular damage and the immune response when we give such high doses of 12 gray. Therefore, vascular damage contributes to the total chronogenic cell kill in tumors irradiated with large single doses of irradiation. So when we are giving very high doses of radiation in saber range, you can have cellular and molecular effects, which can result in DNA damage mediated targeted effects or non-targeted effects. The DNA damage mediated targeted effects can result in apoptosis, mitotic catastrophe, senescence, autophagy, all of which lead to tumor cell elimination. The non-targeted effects can be due to increased production of various neoantigens, increased release of cytokines such as IL-6, IL-8, and TNF-alpha, which ultimately lead to immune cell activation, resulting in tumor cell elimination. Apart from the vascular damage, there is another important mechanism of cell death with high doses of radiation, which is called as radiation-induced immunologic effect. Radiation has immune-mediatory effect on tumors when given at very high doses, and this is called as immunogenic cell death. This is because of the release of cancer-associated neoantigens, which are called as DAMPs, produced by that of the dying cells. There is also secretion of interferon 1 alpha for the recruitment of the dendritic cells, increased expression of CD8 plus T cells, all of which facilitate the homing of effector T cell chemokines and vascular adhesion molecules within the tumor. Apart from it, there is increased expression of CD68 plus tumor-associated macrophages and upregulation of the MHC class 1. Taken together, all these proteins are responsible for immunogenic cell death when we give very high doses of radiation in cellular range. Last but not the least, high doses of radiation can also cause tumor cell death by abscopal. And there are two mechanistic explanations to this. One is the cytokine activation and augmented tumor immune surveillance, leading to tumor growth inhibition, which is basically cytostatic in nature, and then T cell mediated immune activation, leading to cell death, which is basically cytotoxic in nature. So that is the biology of that of the tumor when we are giving high doses of radiation in saber range. But I've also spoken before that when we are giving high doses of radiation to these spine mets, have an adjacent critical structure which is called as the spinal cord. Therefore, advantages of any ablative doses for tumor control in spinal mets must be balanced by minimizing the late effects on the spinal cord. So what is the molecular and the cellular responses apart from the histological changes that we see in irradiated spinal cord? So it must be considered that the histopathological changes within the spinal cord when exposed to high doses of radiation is a distinct process, and that happens due to underlying molecular and cellular responses, which is a continuous process. And this can be in the form of altered gene expression, neuroinflammation, and oxidative stress, resulting in tumor cell eradication. Histopathologically, at the tissue level, what happens is that there is continuous demyelination, vasculopathy, breakdown of that of the blood spinal cord barrier, and ultimately leading to white matter necrosis. This is a simple photomicrograph showing the histopathological changes in the rat spinal cord, which has been exposed to radiation doses of more than 20 gray, which is in the saber range. On the left-hand side is the control group where the spinal cord has not been irradiated. On the right-hand side, it has been irradiated to doses of 2 gray. What you see is in the H&E stain is the white matter necrosis over here, which can be even better seen when you stain this spinal cord with Luxol blue, which is nothing but a stain for myelin, wherein you can see much more profound white matter necrosis. Apart from that, you can also see the disruption of the BSCB or the blood spinal cord barrier using the horseradish peroxidase as a vascular tracer. So when we give such high doses to the high doses of radiation to the spinal cord, there will definitely be damage. Therefore, it is important and pertinent for the purpose of planning SBRT spine that we respect the spinal cord tolerance. To this effect, there has been a recent paper by Arjun Segal's group, which has been published in Red Journal recently, uh, giving a pragmatic dose constraints to the spinal cord whenever we are attempting SBRT to the other spine. And this is for the first time we have modeled these dose constraints based on large clinical data sets. And I'm sure further lectures will be taken on this particular aspect of spinal cord dose constraints by other presenters who will be talking about treatment planning and plan evaluation for SABR of the spine. Therefore, to summarize, the role of 5 Rs is limited in SABR. Double-stranded DNA break is one of the mechanisms of SABR, although not the most important mechanism. It is the cause of direct cell death. However, what is major and is important for SABR is the vascular damage, which occurs due to endothelial cell death by apoptosis and death of cancer stem cells, which reside in the perivascular niche. Other plausible theories for tumor response in SABR of the spine are because of the immunostimulatory or immunomodulatory effect of high doses of radiation, and abscopal effect and bystander effect, which are the major causes of indirect cell death when we are giving SABR. Thank you very much.